Hi everyone, this is Azam presenting on behalf of Tmunity at the Cell and Gene Mesa on the Meeting virtual event in 2020. Our forward-looking statement. And I'd like the opportunity to talk a little bit about our company, which is a next generation T-cell engineering company. We are a private stage biotech formed out of the foundations of the University of Pennsylvania back in 2015. It's a company that's grown a lot since its inception. Now 75 great employees with a lot of cell and gene experience, $231 million raised to date. Unique in that we have our own proprietary manufacturing and R&D center, whereby I'll talk a little bit later on. We are looking to produce viral vector this year, GMP grade for clinical trial purposes and research purposes, and we're well on track to have fully functioning cell manufacturing up and running in 2021. A company with a very large and sizable portfolio, now de-risked further by clinical efforts that have been undertaken and preclinical work, and I'd love an opportunity to share a few thoughts around how our progress is. Despite the challenges of COVID, I am really grateful and very proud of my team who've done an amazing job to ensure the company's milestones remain fairly on track, although clinical trial timelines, as with all companies, have been hit as a result of clinical holds with COVID. So the company's core competencies. We're a company that's driven very much by developing these therapies, particularly with a focus on solid tumors. And our real expertise resides in the fact that we have the real foundations of a great ability in synthetic biology and translation into early clinical settings with our colleagues at the University of Pennsylvania. We developed a very unique model that you've heard me speak about before, whereby the University of Pennsylvania conducts a lot of our early research, discovery, and early translation with a unique model whereby we then work closely with those colleagues to develop IND packages and subsequently file those with the FDA. In certain cases, there are INDs that are held at Penn. In certain settings, there are INDs that are developed within Tmunity. And very early on, within the development cycle, we are thinking very long and hard about the aspects of the product that need to be made right, even in an early research discovery up to translational phase. We think about the tumor microenvironment. We're very passionate about how do we disrupt that with a view to optimizing the targeting, optimizing the signaling, overcoming immunosuppression, and also how do we get the innate immune system to do what it needs to do in terms of tackling the challenges of the tumor microenvironment. I'll talk a little bit about some of these components in the time we have today to show you how this is translated into our products. Our pipeline is very heavily focused on solid tumors. As you can see, I'll talk about two programs today. Our lead program in prostate cancer, and I'll talk a little bit about our approach to advancing the efforts within abnormal glycosylation targeting TN Mukwon in a CAR-T program as well. And our ambitions are that we wish to have an IND filed every year moving forward, and our preclinical pipeline is progressing well despite the challenges with COVID, and I hope to share more details around that in the future. Lead cancer, prostate cancer indication, metastatic castrate-resistant prostate cancer being the lead indication, huge area of unmet need, a need desperately for these patients who have tried all forms of therapies, chemotherapies, radiotherapies. They've had surgical castration, medical castration, and really they now have breakthrough disease with rising PSA levels. We developed a very unique approach to targeting this cancer by looking at the microenvironment and really tapping into what really is a driver within that microenvironment. And we've identified TGF-beta, transformation growth factor beta, as something that we want to target as well as the cancer antigen. And this was elegantly described in terms of a body of work developed by Carl June and Chris Kloss at the University of Pennsylvania. The foundations of this led to an IND, which I'm pleased to inform is progressing well now with dosing in patients in our phase 1 trial. And basically the construct of the payload that we deliver to the T cell includes both a CAR targeted towards PSMA, and you see this as a 4-1BB CAR that we developed, and at the same time we express a TGF-beta dominant negative receptor, transformation growth factor beta dominant negative receptor. And I'll describe in the next couple of slides what that actually means. And basically we're trying to prevent the immunosuppression of these T cells by inhibiting the effects of TGF-beta locally in the microenvironment. So what you see here is a graphic of a normal T cell expressing the TGF-beta receptors. You can see that there's a receptor arm 1 and 2 that are linked together in a helix. TGF-beta binds, and a signal is sent canonically via the SMAD mechanism, SMAD3 and 4, to allow TGF-beta signaling, and in effect that dumbs down the T cells. And what our founders discovered was there was a way to actually truncate one of the receptor arms. TGF-beta dominant negative receptor contains 1 and 2. The receptor 2 arm was shortened and truncated and sequenced and inserted in as a payload, as I mentioned before. So at the same time as the CAR being expressed, the dominant negative receptor is expressed. And this allows the TGF-beta to bind locally at the level of the tumor, but it prevents that signal. But wherever you need TGF-beta systemically, for example, in the renal system, pulmonary or cardiovascular systems, TGF-beta is untouched. So a very unique example of synthetic biology really translating to something in the clinic. And to our knowledge, this is the first mechanism whereby a CAR and both a checkpoint concurrently within the cell is being tackled. Here are some data from the early cohorts that were read out by an original IND that the University of Pennsylvania submitted and developed data, and you can see that we saw on the left-hand side kinetics, which actually, interestingly enough, when you compare them and look to the right, where you see a comparison to CTL019, which is now known as Kymriah, actually very similar in terms of these profiles of kinetics that have been demonstrated. 3 cohorts have been dosed, and we see some quite interesting results with early peak and rise, and then some prolongation that we saw in terms of evidence of some persistence and durability happening in the early time frame. And again, you can see that these kinetics, again, were quite remarkable when you look at some of the higher doses that the Penn colleagues dosed. And this formed the basis of a signal that we saw with an individual that was previously presented in previous MESA meetings and other scientific meetings. And here's a patient who was a 72-year-old male who had PSA levels that were breaking through. Classic picture for somebody with metastatic castrate-resistant prostate cancer. You can see on the curve on the right, the gray bars indicate the PSA trend. Upon the infusion of the CAR-T therapy, we saw a rapid decline in the PSA levels down to the point where they were unobservable. At the same time, we saw a rise in ferritin level. As you know, this is a marker of disease burden in terms of cytokine release. This patient actually had significant CRS, which resolved, and the patient made a good recovery. Unfortunately, a few days later, the patient developed enterococcal septicemia, which may have been secondary to the CRS. We were unable to verify that, and unfortunately, the patient demised. But despite that, the program went on, and we are now in the clinic with this program, dosing patients now and risk mitigating in terms of CRS. And I'm really excited about the progress the company has made on this front. We are now planning to be in 6 trial centers in the United States before year end. We are currently aggressively recruiting patients after COVID hits, but I'm very, very proud of the efforts of the team and the PIs at the sites that have now been very motivated to try and do the best they can for these patients. And I'm hoping to share good news in the future as we continue to enroll patients in this program. Our next target I'm going to briefly talk about is the Mukwon target. This is targeting abnormal sugars in a basket trial, some very sizable indications and unmet need here, as you can see. We've dosed our first patients in the first cohort. We're now into the second cohort and dosing patients in a number of clinical trial centers. Again, ambitions are to have at least additional 6 trial sites open for a total of 8 by year end. Here we're targeting abnormal sugars. On the left, you see an example of a normal sugar chain that you would see, for example, in mucinous cells in the stomach. You see a normal fern tree-like appearance of a peptide backbone with glycan chains, carbohydrate chains, which is a normal appearance. And in cancer, certain types of cancer, for example, ovarian cancer, this normal glycosylation gets hindered, and you get this undernourished kind of stumpy Christmas tree kind of appearance of the sugars. And the reason this happens, we think, is as a result of cosmic mutations, which lead to these very abnormal glycan chains being built. And we were able to discover a unique epitope as a result of this abnormal sugar chain formation, and that is the actual epitope known as Mukwon. And we were successful in being able to work with colleagues at Penn to develop an antibody that targeted this abnormal epitope, and have taken this forward into the clinic. And I hope to share good news in the future, as we believe it's the first time that anybody's gone into the clinic taking TN-MUC into the clinic and targeting abnormal sugars, which historically have been very different. Back in late 19, we were the first company to support work with colleagues at Penn to actually CRISPR edit cells via multiplexing with CRISPR and actually dose patients in a program targeting NY-ESO1 in melanoma, myeloma, and synovial sarcoma. That platform allowed us to really show that we knew how to gene edit in T cells, that we were able to generate human safety data ahead of others. And that has built on our basis now that we feel very confident about our capabilities to be able to manufacture allogeneic therapies in the future as well, with the know-how of what we did in this program. As I mentioned earlier on, our manufacturing has been a core part of our thesis for the company. We started that effort back in 2018 when we leased out our own proprietary facility in East Norriton, which is in the northwest of Philadelphia in one of the suburbs there in East Norriton. We've progressed very nicely in terms of developing great capabilities in terms of analytical development, process development, viral vector development, fully integrated now with a view to getting our GMP vector capabilities in-house up and running this year. Despite the challenges in COVID, I've been really impressed with my team and the commitment and dedication to get that done. And then we are looking at the time period of 21 to be ready for cell manufacturing while still maintaining our great abilities to manufacture at the University of Pennsylvania. And with a real focus on how do we develop a process, or processes, should I say, that radically reduce the cost of goods and are able to scale, because we believe you can firmly do that in the autologous setting with a beachhead forming, and then leverage and that potential to move into allogeneic therapies in the future. We have a great group of investors, raised $231 million to date. You may have seen that late last year we did a Series B and welcomed Andreessen Horowitz to our board. It's a great board to work with. They've been highly motivated in focusing this company on delivering the next wave of generation of T-cell products. It's a privilege to work with all of them. Also welcomed the American Cancer Society on board as an investor in the last round that we did. So we've done some major milestones over the past year in 2019 as we moved into 2020. As you can see, we've demonstrated our first in human data from the PSMA trial ongoing. We've dosed patients in the TN-MUC program. We developed our data in the CRISPR-based TCR-based platform. And also we're well on track now to get our vector needs up and running and GMP scalability for manufacturing within the 2020 timeframe. So this is the remainder of the year will be very much focused on getting our milestones achieved in terms of clinical data, driving the completion of manufacturing, and really advancing our programs, and hopefully advancing and filing another IND in the 2021 timeframe. So our mission remains to be the recognized world leader in T-cell engineering therapies when it comes to focus on solid tumors. We developed a full vertically integrated capability very early on, may I say, in the creation of the company. We believe that this was a critical driver and will be a critical driver for our success in the future. We have a very, very rich pipeline that we are prosecuting against with a broad number of tumors, but a disproportionate focus on solid tumors. We have a unique engine that we've developed, one of a class, best in class, we believe, with our researchers and colleagues, at the same time complementing with significant build that we've done in research and translation, clinical operations, clinical development, as well as the manufacturing and CMC end and quality. And I'm privileged to have a great team that allows me to be the voice piece for it, with some amazing experiences by the founders and management when it comes to T-cell engineering and products. So thank you for your time, and I look forward to being able to discuss more data in the future with you over the course of the next 3 to 4 quarters.